from across the country and actually from across the world. We have international participants as well as local participants. I think, Erin, if you want to move to the next slide, that would be awesome. So it's a real pleasure to sponsor this seminar series from our office, the Office of Data Science Strategy. Our goal is to support a modernized, integrated, and fair biomedical data ecosystem. I invite you to review our website listed here so that you can see all of the work, the opportunities, and initiatives that are supported by our office and support a trans NIH data science ecosystem. Erin, if you wouldn't mind moving to the next slide. So happy Love Your Data Week. This is the last day of Love Your Data Week. As you know, NIH supports fair data sharing and data reuse. And in celebration of Love Your Data Week, we are launching this new seminar series. This will give researchers the opportunity to share their success stories around data sharing and data reuse. This is a monthly seminar series where we highlight NIH institutes and their activities and their programs and their initiatives to support data sharing and data reuse. I just want to note that you can post your questions on Slido. The QR code and the hashtag is listed here. You'll see our welcome note, and then you'll see a lot of activity around where people are coming from. Today's speaker is funded and supported by the National Institute on Drug Abuse, or NIAID. It's a pleasure to introduce my colleagues from NIAID, Dr. Susan Wright and her colleagues. Dr. Wright is the NIAID coordinator for big data and computational science. She has a background in both neuroscience and computer science with theoretical, computational, and clinical research. She oversees activities on leveraging data science to advance NIAID's strategic plan and implement their research portfolios. And so thank you so much, Susan, for introducing our speaker today, and thank you all for joining us.
Thank you, Susan. So good afternoon, and thank you for the opportunity to give an overview of NIDA data science. I'm excited to talk to you about our big data vision and funding opportunities in this area. So as Susan mentioned, I'm the program director for big data and computational science, and I'm in the division of neuroscience and behavior at NIDA, which is the National Institute on Drug Abuse. My contact information is listed on this first slide if you'd like to follow up with me after or have please. So at NIDA, our mission is to advance science on the causes and consequences of drug use and addiction, and we are the primary federal agency supporting scientific research in this area. Addiction is a complex disease that involves neuropathological, psychiatric, and environmental determinants. Like many other fields of biomedical research, a substantial amount of data of various types is being produced, including imaging, genetics, physiological data, and electronic health records. There is an untapped power of data emerging from these studies that lies in their integration, mining, and analysis. We encourage intra and inter-university and multidisciplinary collaborations for data science projects. Next slide, please. Data science at NIDA is a cross-cutting program, and there are 5 main areas of focus for us, which are the integration of existing data sets and tools with those that are being newly developed, making data sets and resources findable, accessible, interoperable, and reusable, or FAIR, ensuring that the algorithms and tools being developed in academia meet the industry standards, data storage and management, and enacting appropriate policies to promote stewardship and sustainability. This approach allows for increased opportunities for secondary data analysis to answer new research questions, as well as to examine alternative approaches for the original questions of previous studies, offering cost and time benefits. Next slide, please. NIDA's vision for how big data science can be leveraged to reveal new aspects of addiction biology is closely aligned with the NIH strategic plan for data science. Areas of interest are data infrastructure, data ecosystem, tools and analytics, community engagement, and workforce development. Next slide, please. To carry out these goals, NIDA is continuously creating opportunities for the latest computational capabilities to be combined with biomedical research. The NIDA website lists the latest big data funding opportunities, which I encourage you to check out for more details, including relevant dates, whether or not a letter of intent is required, etc. I'll briefly mention our funding opportunities now, and please reach out with any questions or follow up with anything specific. So this first list is our notices and notices, or notices of special interest. I'm not going to go through all of them, but I just want to point out that NIDA is participating in the NIH Smart Health and Biomedical Research in the era of intelligence and advanced data science. And also we do have a newly published NOCI on high priority interest to enhance data science research training and addiction research. Next slide, please. This is our list of our current data science research project grants. As you can see, there's several on using existing data. We have signed on to the Biomedical Data Repository and Biomedical Knowledge Bases ones at NIH. And there's also several on leveraging big data science to elucidate the mechanisms of HIV activity and interaction, as well as participating in the CRCNS program. Next slide, please. And finally, I'd just like to mention a few of our upcoming events. We are planning a data science career seminar series that will take place in March and April. The goal of this seminar series is to highlight the career paths of prominent data scientists and NIDA-funded grantees who incorporate data science into their research and hopefully inspire a new generation of data scientists to focus on addiction. Also, we will have a symposium on big data science approaches to identify novel behavior and biological mechanisms of addiction at the upcoming College on Problems of Drug Dependence virtual meeting that will take place in June. Our website also has an archive of past events and recordings if available. And thank you, and please follow up if you have any questions. Thanks.
Thank you so much, Susan. I believe, are you going to introduce Dr. Poldrack? Or Stephen, I think you'll be introducing Dr. Poldrack, correct?
That is correct.
Thank you so much.
So it is my great pleasure to introduce today's speaker, Russ Poldrack. He is currently the Albert Ray Lang Professor, Department of Psychology in Stanford University. Russ got his degree in 1995 from the University of Illinois in cognitive neuroscience, or cognitive psychology. He did a postdoc at Stanford, followed by appointments at some of the major brain imaging facilities in the world, including Massachusetts General Hospital, UCLA, University of Texas at Austin, and then in 2014, he moved to Stanford University. Russ is known for his prolific work in the area of cognitive neuroscience, particularly in the area of cognitive control. But more recently, and more germane to his talk today, he has been a leader in promoting tools and resources for establishing reproducibility and data sharing for brain imaging data. He founded the Open Neuro website and repository, which is a place for researchers to deposit and share data across imaging modalities, including MRI, magnetic MEG, and EEG. There's nearly 500 data sets in this repository and 16,000 participants. He's also been heavily involved in the establishment of a standardized format for sharing brain imaging data called the BIDS data format, the brain imaging data structure. And this will be the topics of his talk today. I just want to put a personal note in. I met Russ at a cognitive neuroscience meeting many years ago when he had first come to UCLA, and dragged him into the substance abuse field, which he has continued to show an interest in, and he currently serves on the advisory board of the Adolescent Brain and Cognitive Development Study. It is my great pleasure then to introduce to you Dr. Russell Poldrack, the speaker for today's talk.
Many thanks, Steve. Let me get my slides going here. Oh, no, that's not what I wanted to do. Let's see. Let me try that. And then, okay, should be seeing my slides now. So thanks very much for the invitation, and thanks, Steve, for the very kind introduction. I want to talk today about an ecosystem that's emerged over the last decade or so for data sharing in human neuroscience. And, I probably don't need to kind of convince anyone here why, data sharing is important. I'll just say that, I think openness is a critical element in general for reproducible science. That's part of why I got into data sharing. The other part is, we wanted to be able to ask questions that we couldn't ask only with the data sets that we had in our lab. There's a great quote from an older paper by David Donahoe that sort of lays out the argument for this. An article about a computational result is advertising, not scholarship. The actual scholarship is the full software environment code and data that produce the result. And so that sort of philosophy motivates a lot of the work that we do. So I want to talk about a set of kind of interlocking things that my lab has been working on for a while now in service of reproducibility and open science. And the first is open data. We've been involved in a number of different projects that share data, and I'm going to focus particularly on Open Neuro, which Steve mentioned, but we've also been involved in NeuroVault and NeuroSynth. We also develop a number of open tools for reproducible data analysis, and I'll mention MRIQC and fMRI-Prep. And then finally, all of this is sort of undergirded by open standards, particularly the BID standard that Steve mentioned that I'll say more about. So it's interesting that our field actually had a bit of a false start back around 2000 when this project was developed called the fMRI Data Center that was basically meant to be a platform for sharing data. It was a really, sort of forward-thinking idea, but sort of, I think, made the mistake of trying to push too hard when the field wasn't ready. And in particular, a number of journals started requiring submission of data to the data center in order to publish the papers. And this led to a bit of an outcry. In 2000, there were, sort of commentaries in Nature Neuroscience. There was an open letter that was sent around by a number of, pretty prolific scientists from the fMRI world. That I'll just read this last little bit. We're particularly concerned with any journal's decision to require all authors of all papers accepted for publication to submit all experimental data pertaining to their paper to the data center. Now that kind of requirement doesn't really sound revolutionary now, but in 2000 it was not something that people had sort of gotten their head around. Now, interestingly, our field has really come around, and really it was over the last decade, particularly arising from the work of Mike Millam and Cameron Craddock in this INDIE project that really sort of drove the rebirth of data sharing. And so this is from a paper that we wrote a few years ago, highlighting that there's this whole, what we refer to as an ecosystem that's built around kind of different types of sharing. Sometimes you need the raw data, right? Sharing raw data is hard. Some things you can do with much more simplified data sets where it's easier to share them, but there's kind of less information there. And so there's projects working at each of those levels. It's interesting. This is a quote from an anonymous senior researcher overheard a year or two ago at the Human Brain Mapping meeting, saying that OHBM has been taken over by the open science zealots. So that's the degree to which I think the energy in our field now is really behind data sharing and open science. Now, a lot of effort and attention has gone to projects that you could think of as prospective data sharing projects, where the goal of the project is to collect a data set that will be shared. And so, really prime examples are the Human Connectome Project, which the first kind of young adult Human Connectome Project finished a few years ago, shared data from about 1,200 individuals, and it's been used in a very large number of publications, but it's by far the most analyzed data set in the brain imaging world right now. You're probably familiar with the ABCD study, which has the aim of collecting data longitudinally from roughly 10,000 children. So we focused in our work on what you might think of as retrospective data sharing. This is really kind of sharing of data that have been collected in labs for the purposes of a particular study. And you can think of this ecosystem as sort of falling along these dimensions of breadth and depth, right? So at the one end, you have databases like NeuroSynth, which stores basically coordinates of activations that have been extracted automatically from papers. So this is something that allows you to do it automatically. So this database now I think has on the order of 14,000 papers worth of data in it. But you're limited in what those data can tell you because of the nature of the data. On the other end, we have Open Neuro, which shares fully open raw data sets. much more information there, but much harder to share because somebody actually has to organize the data and submit them. And then in the middle, I'm not going to say much today about NeuroVault, but NeuroVault's a project that tries to kind of sit somewhere between there, which allows people to upload the kind of derived results at a whole brain level. So for example, a whole brain statistical map and allows visualization of those sorts of maps. So there's this ecosystem that kind of varies. I'm going to focus really on the raw data side today because I think that ultimately that's probably the most useful for lots of questions. But the one of the main points that one has to appreciate is that simply sharing data is not sufficient. You need to share it in a way that actually makes it useful. And if you haven't seen it, I would suggest, I'm not going to play it, but suggest checking out this video on YouTube, Data Sharing and Management Snafu in 3 Short Acts. And this is just a little extract where it's basically a person who wants some data talking to a researcher, trying to get the data from them. They finally convince them to send the data, and this is the person speaking. I received the data, but when I opened it up, it was in hexadecimal. The researcher, yes, that's right. I cannot read hexadecimal. You asked for my data and I gave it to you. I have done what you asked, right? So that's kind of reflective of a philosophy that I think at least some people have had in the past. Is there a guide to the data anywhere? Yes, of course. It's the article that is published in Science. Now we all realize how ludicrous that is. So we started working a number of years ago on a project to basically develop a standard for organizing neuroimaging data, and it's come to be called the Brain Imaging Data Structure, or BIDS. And it really comprises 2 parts. One is a file organization standard. How do you name your files and your folders? The other is a metadata standard. How do you organize the metadata that's associated with the imaging data? So this grew out of, so before OpenNeuro, we had a previous project called OpenFMRI that focused more on kind of explicitly on task fMRI. And it had a standard for kind of how one organized one's data, but it was just kind of ad hoc. So we decided we wanted to bring together the community to really come up with a standard that would meet the needs of a much wider range of researchers. So we had initial meetings in 2015, funded by INCF, and developed a draft standard that we sort of posted to the BIDS website along with a bunch of example data sets later that year. So basically sent it around to the community for feedback, and then in 2016 published the paper and also published the first first official standard. So BIDS was really, the idea was really the brainchild of Chris Gorgolewski, who was a researcher in my lab for a number of years, moved to Google a couple of years ago. And so it's been an interesting transition because, he really was the leader of the project. And there was a lot of concern that when he left academia, that the project would just fall apart. But with the work of a lot of people, it's actually continued to grow. These are images from the groups who were involved in sort of early work in developing the standard, meetings held at Stanford and Princeton. But you can now see, if you look at GitHub, to the BID standard organization, and look at the specification, there's been 60 contributors. We have 4 maintainers who, really sort of put a lot of work into making things happen. And then we have an elected steering group that I'm part of, and you can see just how international it is. And it also has people working in many different imaging modalities, from electrophysiology to fMRI to structural MRI to PET. And so I think that, that it's really grown into a really impressive community-based standard. So we started with a set of principles in BIDS that sort of reflected, in part our experience in sort of previous informatics projects. The first was that, the thing we care most about is adoption. Like if we don't want to build something if nobody's going to use it. And so one of the things we try to do that is to keep the standard as close as possible to what people already did in their lab. And that often meant not using things that were technologically necessarily the best option, but we didn't want to let technology override usability, right? There might well be a file format, for example, that's like a better file format, but if nobody in our field knows how to use it, it's going to impede adoption. We also try to focus heavily on engaging the community, and I'll talk more about how we try to kind of incentivize adoption in a bit. The second was, don't reinvent the wheel. Whenever there was an existing standard or file format, we tried to use that. Now, we couldn't always do that, but that's what we tried to do. And the third is, what I refer to as the 80-20 rule, which is really kind of not letting the perfect be the enemy of the good, right? Not letting sort of edge cases drive over engineering, or drive a kind of paralysis, by trying to deal with things that may well not even come up. So we really focused on the use cases that could, allow most people in the field to take advantage of the tool. So when we get data off of an MRI scanner, it usually comes in the DICOM format, which is pretty useless for researchers, in part because the organization doesn't really reflect the way that we think about, how data are organized. So what we did in BIDS was basically think about what's the structure that most researchers use, right? Well, I usually have a data set. I'll have some metadata about the participants in the study, and then I'll have data for each individual subject. And that for an fMRI study, that might include, anatomical images, functional MRI images, diffusion images. And so we built a framework that sort of looked like the way that people were often already doing this in their labs. So it's not a huge lift to move from existing organizations to BIDS. So just to look at, a few of the types of formats that we use. For image formats, we standardized on the NIfTI format, which has been around for about 20 years now, and is really accommodated by pretty much every imaging analysis software, at least in the MRI world. In electrophysiology, there's been other formats that are being used as well. We try to keep things as simple as possible. So for metadata, when it makes sense, we use tab separated value files, tab delimited text files. For data where we need more of sort of a dictionary format, we use JSON files, which is again a file format that most people are able to deal with pretty easily. We have two different templates. One is a directory naming template, which basically says, here's a guide to how to name directories, and there's options at various levels. Sometimes, for example, if you have multiple sessions, you might want separate directories for different sessions. Sometimes you don't have that, so you don't need it. And then probably the most important thing is the file naming template, which basically provides a way to embed, metadata, the most important metadata about the file, to embed that in the file name. And again, that's, there are many people who don't like to embed metadata in the file name, but this is generally what researchers do and what they expect. And so that's why we stuck with this. Now, when we had built the earlier standard for OpenFMRI, basically the only way that we could tell whether a data set matched the standard was basically to try to run our analyses on it and see whether they crashed. And that's not a very useful way to tell whether something is standards compliant. And so when we started BIDS, we really focused on the idea that we have to be able to validate a data set easily. And in particular, because BIDS was really built with OpenNeuro in mind, and we wanted to be able to automatically ingest data. We want to basically allow somebody to click on a data set and say, upload this. And before it gets uploaded, be able to look at it and say, hey, either this passes the BIDS standard, it's a valid BIDS data set, or it doesn't. And here's some guidelines as to what's wrong with it. So we developed a JavaScript validator. And this is, you could run it, in the browser or on the command line. This is a kind of a view of what the output looks like. So, it basically very quickly, I mean, even for a data set of like, hundreds of gigabytes, it can validate it in, a number of seconds. And so because it's just looking at basically file naming and file structure and metadata, it's not actually looking inside the imaging data files. So this, it tells you basically this is not a valid data set. If you would be able to unfold this error message and see, what are all the errors, so you know how to fix them. So BIDS started focused on, raw structural, functional, and diffusion MRI data, but has since been, we now have a process in place that's kind of based on the way that this works in open source software projects, to basically propose what we call BEPS, BIDS extension proposals. And so we've already had a number of those. There are extensions to MEG, EEG, and intracranial EEG that have been sort of finalized. There's also one that's been finalized for derivative data, that is data that have been sort of processed. So even though we started with raw data, now we have at least a beginning standard for what we call common derivatives. We're just about to incorporate standards for PET for quantitative MRI and for arterial spin labeling MRI. And there's also one for eye tracking. There's a number of others as well. These are the ones that are sort of fairly far along. And then there's a number of them for different data types like connectivity matrices, statistical models, image transformations. All of those are sort of ongoing right now. But the important point is that anybody can propose an extension. And then in general, what would happen is, they'll write it up, and then at some point they'll come talk to the steering committee and we'll give them feedback about it. The BIDS has been pretty widely adopted. It's been endorsed by INCF as it was actually the first community standard that they endorsed. That 100 labs number is a couple of years old. It's being used in probably hundreds of labs at this point. Been adopted by a number of projects. The ABCD data have been converted to BIDS, and I don't know if they're actually available on NDA yet, but if not, they're coming. And there's well over probably that number is probably 30,000 or 40,000 at this point, anonymized data available that are formatted in BIDS. Now, I mentioned that we wanted to get people to adopt BIDS, and one of the things that we wanted to do was basically say, if you move your data into BIDS, here's a set of tools that you can easily apply to your data. So as an example, any of you who've there's a program called FreeSurfer that's commonly used for doing these sort of, very fancy reconstructions of the cortical surface. And it has its own, special file organization structure. In general, if you want to run FreeSurfer, you need to sort of put your data into that structure. We built a BIDS app, what we call a BIDS app, which is basically a containerized software application that has sort of a layer, kind of a compatibility layer that allows the software to understand the BIDS data structure. So that now if you want to run FreeSurfer on your data and your data are in BIDS, you just point this BIDS app at your data set and it will run without you needing to sort of reorganize anything. In addition, because it's containerized, we generally use Docker for this, or Singularity, that container encapsulates all the software and all the dependencies. So you can, as long as you know the version of the container that was used, you can exactly reproduce the analysis down to, kind of floating point weirdness. So we laid out this kind of idea for BIDS apps, and there's now probably 30 or 40 BIDS apps available. I'm going to mention a couple that my lab has been involved in developing. One is called MRI QC, which is a quality control app. Quality control has always been, a bit of a hairball for researchers in MRI because there's never been any like really easily usable and visualizable tool. So what MRI QC does is allow you to basically, you point it at a BIDS data set and it gives you back a report that kind of looks like what you see at the top here. And then let's say you want to know, like, why does this particular individual have really high CNR? You could click on that and sort of, go in and look at that particular individual's maps and see what might be going on. This was a brainchild of Oscar Esteban, who's now in Lausanne, Switzerland. I'll also mention that, one of the things that we do in both this project and in the project I'm going to mention in a moment is we build in telemetry that people can opt out of, but if they don't opt out, basically every time it's run, it sends some information back to a central system, anonymized information, no information about the images themselves. But in this case, the image quality metrics are sent to a database that's run by Adam Thomas and his colleagues at NIMH. And so basically we now have quality control metrics from almost 300,000 unique fMRI scans and over 250,000 unique T1 weighted structural images. So for example, this is just one analysis done a while ago. So, if you want to know, how does the temporal SNR relate to multiband acceleration factor? This is, plot of a bunch of different data sets at different acceleration factors showing you the TSNR. And we published a data descriptor in Scientific Data describing, anybody can access these QC data. Another tool that we've been involved in developing is something called fMRI Prep, which basically tries to build a very robust and reproducible pipeline for fMRI pre-processing, right? So this is a schematic of the fMRI pre-processing pipeline, which is pretty complex. There's lots of steps. And most labs really vary in how they do this. There hasn't been kind of a single kind of agreed upon pipeline. And often, people they're not doing it in a way that's optimized. They're just kind of taking whatever the first postdoc in the lab, put together to do it. And so we wanted to build something that was really robust, that could take advantage of whatever information, whatever images are in the data set, in order to be able to do the best possible job, to do it reproducibly, and to do it robustly. And so fMRI Prep does that. It's another BIDS app. This is just an example of the kind of reports that it gives. So one of the challenges with this kind of software is, you're in some ways, you're just offering people a black box that kind of takes in data and gives them out results. And you want them to be able to really understand whether those results are trustworthy. And so one of the things we have what we call a glass box philosophy, which is that we try to open up the black box to show what's going on the inside. So this is just one example of the documentation, the report that comes out showing, here's the workflow that was applied, here's some results. And then we've developed some particular types of visualizations. This is visualizing the registration of functional to structural images with the cortical surface overlaid. So you can kind of see them flipping back and forth. And it provides a really easy way to tell if something has gone wrong visually. I mentioned that we get telemetry back to tell us, importantly for fMRI Prep, to tell us how often is it being run and how often does it actually succeed. So this is from the last couple of months of 2020 and the first month of 2021, showing the number of successful runs per week. And you can see that it's being run at least 2,500 times successfully. Right now it has a success rate of about 80%, so 80 to 90%. So so what we can do then is we also, when it crashes, we can get some information back about what step it's crashing at and what sort of errors are happening that lets us try to head off problems. I'll also mention that one of the things that we've done recently with fMRI Prep is generated what we call a long-term support version, and this is really meant for projects that are going to have to be processing data over many years. And what we want to do is basically give a version where we can guarantee that it'll continue working. So we'll fix critical bugs, but we won't actually do anything that changes the processing stream. And that's meant to, to really provide a longer term alternative for, sort of projects that need longitudinal stability. So let me talk now about OpenNeuro, which is the platform that we've developed in our group for sharing of a broad range of imaging data. So basically the idea behind OpenNeuro is any data for which, basically any data set that can pass the BIDS validator can be shared on OpenNeuro. And so right now that means MRI, MEG, EEG, and intracranial EEG data. So this is what you see if you go to openneuro.org. We can see we have almost 500 data sets, more than 15,000 participants. When you click on, if you just go to the public dashboard at the top, you don't even have to log in to see all of these data sets that are available. These are, I think, sorted by how many stars people have given them. And if let's say you click on this top one, which is this UCLA Consortium on Neuropsychiatric Phenomics data set that we shared a while ago, it's been downloaded 1,400 times. It's had 1.9 million eyeballs on it. And this is what one of the data pages looks like. So the first thing you see is that it tells us basically whether it passed the BIDS validator, which it needs to have in order to to be shared, and whether there are any warnings. You can kind of browse the data if you want, and then there's a bunch of other metadata that are provided. So this is what happens when you first go to upload a data set to OpenNeuro. You select the files, and the first thing it does is it runs validation on your data set. And so then if it passes, you can go ahead and upload it. But it does that before actually uploading the data. So you'll see on the left that there's various versions of the data. So before anything can be shared on OpenNeuro, it has to be basically, we call it snapshotting. You have to create a snapshot of the data set, and that creates a specific version that's immutable. So now if somebody wants to download version 1.0.0, we guarantee that data set will always be exactly the same. It's never going to change. So if one wants to download, there's a number of different options. You see there's the download button here, where one can download the browser. But in general, for larger data sets, we recommend that people either use because the data live on Amazon S3, either use the S3 client. We have an OpenNeuro command line interface, or you can use the DataLad tool, which is basically DataLad is the back end that everything runs on top of for OpenNeuro. If you're not familiar with DataLad, it's a project that basically lets you sort of view data sets, sort of like GitHub repositories. And so that's a really handy way to actually grab a data set. Every snapshot is provided with a DOI that includes, the data set accession number and the version number. So that, and that's, I think that one of the main reasons that people upload their data sets to OpenNeuro is because their journal is requiring them to provide a DOI for the data. And so this is just an example of, one of the papers from James Booth's group that, cited their OpenNeuro data set and cited the DOI. Now we also include a license for the data, and I know that talking about licensing of data is not exactly the right terminology, but that's the terminology that most people think of. And right now, all data shared via OpenNeuro are basically provided using what's called a CC0 or a public domain dedication. That basically means that the work is put out into the public domain and anybody can do basically anything they want with it. There's no intellectual property restrictions. There's no requirements for authorship, no restraint on what you can do with the data. So that's at the end of the distribution of the degree of control that people often want to exert. Now, obviously this requires us to protect our subjects. And one way in which we do that is we recommend that anybody, we provide basically, we don't, but a group who develops something called open brain consent, our community provides language that basically suggests, how to consent individuals to make clear that their data are going to be shared openly and, alert them to what the potential risks are for that, as well as the benefits. Now, obviously, if we're going to share our data and, and claim that it is de-identified, we need to remove any identifying information. And the main thing, in addition to all the standard stuff like names, which is, the BID standard takes care of most of that. One of the main things that we have to worry about in imaging is facial structure, right? We know that if you have a whole head MRI, it is theoretically at least possible to identify an individual from, if you have images of a bunch of people, identify that individual based on reconstructing that image. So we, basically we require researchers to remove the facial structure from any images before they upload them. We provide a tool, others provide a tool as well. Now, right now we have to trust that people will properly do that, and we basically have a curator go in and check. We're currently developing an automated face detection tool that'll be able to look at images basically before they're shared and make sure that there's no facial structure available there. Here's an overview of kind of where the project stands. So we've been funded for a couple of years right now as one of the official data repositories for the BRAIN Initiative. And we're also part of the Amazon Public Datasets project. We have, as I mentioned, almost 500 data sets, more than 15,000 subjects. We get about 10 to 20 new data set uploads per month, and that's been consistent over more than the last year. We serve on the order of about 1,000 plus data set downloads per month, usually on the order of 20 terabytes of data. We have on the order of 7,000, I think it's actually up from that now, users in any particular month. There's a bunch of papers that have been published that are either associated with, basically associated with a data set that's published in the database, or that have actually reused data sets from OpenNeuro. So I want to end by just talking about what I see as some of the keys to success, like why has data sharing succeeded in the field of neuroimaging? One has to do with the data. Like the data are largely digital end to end, right? So it minimized their, they come off the scanner, the MRI scanner, as as digital images, and so it minimizes the manual steps in the process. Obviously, so we're lucky in that sense. We also have standardized file formats and data standards, right? So we have BIDS, we have NIfTI, and this makes the data immediately usable by anybody and really reduces the burden of curation and preparation. So you can think of what we've done in OpenNeuro as basically pushed curation off to the user, right? We basically say your data needs to be in BIDS. We validate it before we upload. So basically the curation is largely, the responsibility of the user. And we did that because curators are expensive. We couldn't afford to hire an army of curators to do a bunch of sort of, data preparation. Another is sort of demonstrated scientific utility, right? And we now have a bunch of success stories of people who've, built careers asking interesting questions using, sometimes only using shared data. I think another lesson that we've learned is that community buy-in is really essential. That if you try to, what the fMRI DC story shows, if you try to put a mandate in place before the community's ready, it can backfire. Unless it's obviously it's worked differently in genomics, but there you had overwhelmingly powerful advocates. I think another thing that's been important is that the people who've been kind of advocating for sharing are, like myself and Mike Mill and many others, are really kind of bona fide members of the research community, not sort of people from outside saying here's something you should do. And as they say in Silicon Valley, we're eating our own dog food, right? We're doing the things that we say people should do. I think another is really keeping it simple and as close to standard practice as possible. So this is an image from a project that was developed more than a decade ago, the Biomedical Informatics Research Network, which tried to basically build an infrastructure for data sharing and pretty much failed. They were able to share some data, but they never got any uptake by more than a few groups. And I think in large part that was because the solution that they built was so over-engineered that nobody nobody could use it. And so one of my heuristics is that if there are more than two acronyms that somebody's never heard of, they're probably going to sort of glaze over when you show them a figure like this and tell them that this is how they have to share their data. So we've tried to basically keep things as close to practice as possible. And finally, we've tried to be really sort of diligent about not letting the perfect be the enemy of the good, and realizing that there's always going to be edge cases, and there's always going to be people who think that if you can't deal with every edge case, you failed. And we basically just let those people keep talking and really focus on covering, doing the effort that'll let us cover most of the data sets. And I think the success of BIDS and OpenNeuro has shown that, at least in our community, that's been a good approach. So finally, just to wrap up, I have this picture on the right because, we do live in a bit of a bubble in our field. Sometimes I talk to people from other fields who are, who are much farther behind where we are in data sharing. And it's hard for us to often remember how hard the last 2 decades have been to get to where we are now. But I think we do have a really enviable ecosystem for open science. And infrastructure has been really critical to ease the friction around making that open science happen. So things like validator-driven data sharing, and containerized apps to encourage adoption of the standards. But really, ultimately, it's come down to community engagement. And I think keeping the tools as close as possible to researcher practice has been really important. So I want to end by thanking, this is what my lab looks like now in the days of Zoom. And in particular, thank Chris Gorgolewski, who's now at Google, who was really sort of key in getting both BIDS and OpenNeuro off the ground, and Squishy Media, who's a contractor who's done all the software development work on the BIDS website and a lot of the work on the validator as well. And then, we've had funding from a number of both NIH and other groups that have allowed us to do this. So thanks very much. And I'm happy to have some discussion now.
That was great. Thank you, Russ. So there's a few questions that have showed up. And I guess the first one, do people outside the neuro and fMRI community use BIDS and NIfTI?
So people broadly in the MRI community use NIfTI beyond, so certainly, beyond fMRI. It's not really used outside of the MRI community. BIDS, is now, as I mentioned, it's being extended to things like electrophysiology, to like EEG, MEG, intracranial EEG, PET, ASL, other things. So it's an extensible standard, and so it's growing, but it's not really being used by people outside the neuroscience community now, and in part, it's not really intended to be, right? It's really intended to be a standard for a particular community, and other people are being inspired by it. For example, there's an emerging standard called PsycDS, which is basically meant to be like BIDS, but for psychology data. And so, sometimes it makes sense to add things to BIDS. Sometimes it makes sense to kind of go start a new standard.
The next question is, does BIDS use bioontologies for controlled vocabularies?
The short answer is right now, no, but there's work going on by Dave Keeter's group at Irvine that's supported by a Brain Initiative grant that's working on sort of integrating ontologies into BID, so that I think in the near future we will be able to link to ontologies.
The next question is, do you connect with other repositories to add different types of data?
Short answer is right now, not in a systematic way. So we're working on linking with the NIMH data archive. And in particular, we want to be able to kind of federate search. So if there's a data set that includes both, something on NDA and something on OpenNeuro, one could easily find those things. And right now people can, in principle, add those links into the description of the data set, but we'd like to do that kind of more directly, but there hasn't been a big outcry for that kind of linking other than wanting to link back to NDA. And that's why we're working with the NDA folks to try to make that happen.
What are some of the benefits for uploading data into the repository, and how would you persuade an investigator, especially in the light of risks for patients?
So I think the benefits, there's a couple of different benefits. One is like getting one's data set seen and cited, right? That what people have seen is that data sets that are shared in general, the papers associated with those data sets, get more citations because people, see and use the data. The other benefits are, being able to be part of larger consortia. So, often there are people who want to combine data sets across multiple studies, and that can kind of get one to actually, even though the OpenNeuro doesn't require that somebody be, even cited or included as a co-author, often that ends up happening because it often makes sense for somebody with expertise in the data set to end up being a co-author. And then finally, journals often want data to be shared. So for example, PLOS One requires a DOI for the data set, and OpenNeuro provides that. So, I don't go out of my way to try to persuade investigators. I feel like, we offer incentives, and I think like the incentives that I just described are often good enough. Obviously, we have to understand that, as the question mentions, in light of the risk for patients, and we try, through anonymization, we try to just kind of minimize those risks. Obviously, one can never completely remove the potential risk for re-identification. For most of the data sets in OpenNeuro, I think so there's two questions, right? There's the probability of re-identification, and then there's the magnitude of the impact on the person if their data are re-identified. For most of the data sets in OpenNeuro, there's relatively little metadata that, even if they were re-identified, they wouldn't really, it's hard to imagine how they would incur risk. Obviously, there's some that have information related to like patient status, and there we just sort of take advantage of the fact that we've done, we've at least kind of lived up to the HIPAA safe harbor requirements in kind of removing everything that we can. And we've had our privacy office at Stanford review the processes, and they're happy with the degree to which that we've done that.
The next question is, why would you use NIfTI and metadata rather than DICOM as image and metadata?
I guess the main reason is that pretty much no software packages that people use for scientific research in the imaging field can work with DICOM data. So pretty much everybody in the field converts their data to NIfTI anyway. The other issue is that DICOM images often contain, or they almost always contain, header metadata that would be considered PHI, with things like times and dates and often names and stuff like that. And so the conversion to NIfTI also helps to to provide a layer where we can sort of remove, get rid of some of that identifiable metadata.
The next question is, how extensible do you think the BIDS model is to other organs, and are other communities ready for the paradigm shift?
I think, in principle, it should be, directly extensible to any kind of, imaging of any other body part. The I don't actually know the answer to, how kind of whether these other communities are ready for the paradigm shift. I haven't had kind of, deep enough discussions with people outside of the neuroimaging community. It's an interesting question. There certainly have been people, interested. For example, there's an ongoing effort to develop a microscopy standard for BIS. Now, whether that will end up being kind of, merged with some other microscopy standard outside of BIDS or not, we don't know yet. But there certainly are, some other communities who are interested in this stuff. But it's an interesting question. I think it certainly in principle should be extensible.
And the next question is, how does BIDS relate to XNAT and other database approaches?
I think the main difference is that, certainly XNAT is meant for grabbing data directly from an MRI scanner, as are things like Flywheel. Whereas, so I would think of it, so they're going to be basically taking in DICOM data and then, potentially outputting data into another format. And BIDS, so I don't know if it's built into XNAT now, but there certainly is a converter where one can take a data set that lives in an XNAT database and output it into a BIDS format. Similarly for Flywheel, if you have data in a Flywheel database, you can output it into a BIDS format. So I think that I view BIDS as really kind of the layer that sits between a database like XNAT or Flywheel and the kind of analysis that's going to be applied to the data, which is usually applied outside of the database.
And then the next question is, have you encountered any issues regarding cloud-related performance and costs, such as data storage or compute for your data sets?
So we sort of. So, one thing to say is that for OpenNeuro, for the databasing itself, because we're part of the Amazon Public Datasets project, they actually provide us with free storage. Now we have to pay for all the compute that it takes to run the site, and that's not a cheap thing to pay for. But in my lab, we don't do much kind of cloud computing for our actual data analyses. So I'm not exactly sure what that question was getting at, but it's certainly it is a challenge. And I'll highlight the fact there's a report coming out from a group that I've been involved in on what's called neuroimaging data in the cloud, which addresses a lot of the questions around sort of using cloud computing for neuroscience data analysis. I think if you just Google neuroimaging data in the cloud, you should be able to find it. It's a National Academies report that'll be coming out soon.
And the next question is, we've received pushback regarding brain image defacing techniques available adversely impacting obtaining accurate brain measurements.
Yeah, there certainly are. Yeah, there certainly are some analyses that one might want to do that are impacted by defacing. In particular, for example, building head models for MEG or EEG really requires facial structure, and that's just that's a problem that I don't think we can deal with in if we're going to share data in this way. And so it may well be that there's that we need an additional layer of data sharing either, and we may end up doing this as part of OpenNeuro in part because we're also going to have to if we want to move to sharing data that are covered by GDPR, which we can't right now, we're going to have some sort of level of authentication and permissions control, and so it may well be that ultimately we have to share those kind of data under more restrictive circumstances. So far we've limited ourselves to only to data that can be shared in this kind of open way.
And several people are curious, what type of computer infrastructure and hardware and software are you using?
There's two ways to answer that question. One is around OpenNeuro. How do we actually share stuff? And the answer there is that we run everything on AWS, on Amazon Web Services. For the analyses that we do in my lab to actually analyze data, we work with either the Stanford Supercomputing Center or the Texas Advanced Computing Center to to basically use high performance computing, because none of this stuff can be done on kind of individual systems. And rather than running my own systems, I prefer to use, campus level or national level supercomputing resources.
And the next question is, how do you make sure investigators submit sufficient metadata that describe the data uploaded? And also kudos on the building standards and a data validator.
Well, thank you for that. So the short answer is we don't. There are certain metadata that's required by the BID standard, which I think is like age and, a few other things around the individuals, and then, things around the data set. But the short answer is we don't. And what we instead do, so if you go to OpenNeuro and you're at a data set and you want to know something about it, there's basically a question section where you can put in a question, and that'll automatically get emailed to, or the owner of the data set will get notified, so they could come try to answer the question. So that's the way that we try to address that. And then, if there's something that's missing that is needed, then if the investigator has it, they can upload it to, you can always upload kind of an addition to a data set or change files in the data set. And then, share an updated version of the data set. That's the way that we've dealt with that issue.
And do you hear concerns around the sharing of bad data? And is it possible to balance the desire for speed with maintaining the quality and accuracy of the data?
So, we've taken a philosophy. So certainly some people raise this issue. We've had the philosophy of, if people have data that they claim that they're using for science, then those data should be shared. And, one person's good data is another person's bad data, right? So we basically feel like if somebody feels that the data are good enough to share, then they should be shared. And it's the responsibility of the reuser of the data to make sure that the data quality is sufficient for their particular purposes, right? Because sometimes, data are perfectly good for one purpose, but not for another. And so we don't see ourselves as the gatekeepers of whether the data are good or bad. And one thing to mention is, we're working on basically running, we don't currently run all of the data sets through MRIQC, but we do, we are planning to basically provide MRIQC reports for all MRI data sets in the OpenNeuro database. And with regard to this kind of speed versus quality accuracy, I don't view that as a repository. I don't view that as our role. I think that our role is to provide people a way to share the data, and they can kind of, make their own decision about that particular trade-off themselves.
Do you have any idea on how to improve the visibility of shared data with potential researchers other than publications that reference the DOI?
Well, so there are increasingly, various search tools. So for example, Google has a data set search tool, and we, try to expose our metadata in a way that gives visibility on the Google data set search tool. And then, as I mentioned, for example, we're working to try to integrate our database with the NIMH data archive, so that somebody who's searching on either database could find data in the other as well. I think that those are the ways that at least we're thinking about it.
And then in addition to DOIs for data sets, are you using identifiers for researchers, samples, equipment, and labs? And are you using usage stats to showcase benefits of reuse?
So the short answer is no, we're not using identifiers for, actually, sorry, we do allow individuals to add their ORCID, researchers to add their ORCID. They're not required to do so, but they can. So that's, so we support that. We don't use identifiers for like samples or subjects. So there is, for example, there's this GUID that's used by the NIMH data archive to identify individuals. We haven't gone down the road of using that. And we're working on, we need better usage stats. We can kind of like, reap stuff on our own on the back end, but it's not easy. And obviously, DOIs provide probably the best way to see benefits of reuse. But that's something that's kind of an ongoing, improvement that we're trying to work on.
Thank you. I think we're out of time for questions, but thank you again. And thank you all for your questions. And I'm going to turn it back to Susan Gregorek.
Thank you so much, Russ and Susan and Stephen, to everybody who joined us today. This has been a really insightful and rich discussion and presentation. I really thank you for your time and efforts. Please join us in March, roughly the same time, it's roughly the same day, when we'll have another presentation on open data sharing and data reuse by, I believe, NHGRI. And with that, I'd like to close the session, and everything will be made available through our website shortly.